By the time you look at a boxer, you think you know the story. A loyal, square-jawed guard dog. But in 2004, a single genetic study shattered that assumption. Researchers didn't just find a dog genome, they uncovered a hidden history that rewrites everything we thought we knew about the breed's origins. Today, we're looking at the DNA secrets that have been hiding in plain sight. That detail changes everything. Tasha was not just another registered dog in a kennel record. In the hands of researchers, she became a reference point, a living map of inherited code. The boxer, a breed already famous for its clear physical identity, suddenly stood at the center of a question that was much larger than appearance. If this one dog could help reveal the architecture of the dog genome, then what else about the breed had been misunderstood for decades? The surprise was not only technical, it was emotional in a quieter way. People tend to trust breeds that look stable. They see a boxer and assume the shape tells the whole truth. Yet the genome of Tasha suggested that behind that familiar outline, there was a deeper history waiting to be read. The breed was no longer just a symbol of strength and discipline. It had become a doorway into ancestry, variation, and the hidden record of selection. Researchers did not begin with a myth. They began with a body, a set of chromosomes, and a dog whose DNA could be examined with unusual clarity. And because that genome was of high quality, the boxer immediately gained a scientific importance that went far beyond its reputation in parks, show rings, or family homes. Tasha's sequence gave scientists a framework for comparing other dogs, tracing inherited traits, and asking which features were ancient and which were shaped more recently by human choice. That is where the real unease starts. A breed thought to be well understood became the unexpected standard for looking into canine origins. The boxer was not simply being studied, it was being used to test assumptions about how breeds are built, how traits survive, and how much of a dog's identity is written in the visible form versus the invisible code. In that sense, Tasha was not just the first boxer genome. She was evidence that the most familiar breeds can still conceal the deepest questions. The Broad Institute's Dog Genome Project turned that single sequencing effort into something larger than a laboratory milestone. It placed the boxer at the front of a scientific conversation about lineage, inherited structure, and the long path from ancestral dogs to the breeds people recognize today. And once that path was visible, the confidence people felt about the boxer's origin began to look less like certainty and more like a carefully maintained assumption. That is the point where the story stops being about one dog and starts becoming about what the dog had been hiding in plain sight. Because if a boxer named Tasha could expose the limits of what people thought they knew, then the next question was unavoidable. What exactly was buried inside the breed's ancestry, waiting for the DNA to make it impossible to ignore? By 2011, the quiet certainty around the boxer's origin had started to crack in a very precise place. Chromosome 26. Researchers working with the boxer genome identified a selective sweep stretching more than 8 millibars, an unusually large stretch of DNA that carried the unmistakable smell of human intervention. This was not a vague hint or a decorative detail. It was the kind of genomic signature that suggests a trait was not left to chance, but pushed, protected, and fixed across generations. And that changed the question immediately. If such a broad region had been swept clean by selection, then something in the boxer's body had mattered enough to breeders to leave a long scar in the code. The visible dog suddenly looked less like a finished natural outcome and more like a record of repeated decisions. A head shape, a profile, a working frame, a face that people recognized at a glance. The DNA was saying that these features had been guarded so closely that variation around them had been narrowed down to a remarkable degree. The paper from 2011, published at https://pmc.ncbi.nlm.nih.gov/articles PMC 3152542, did more than describe a technical anomaly. It pointed toward strong artificial selection for specific characteristics, and one of the clearest possibilities was brachycephaly. That mattered because brachycephaly is not a minor cosmetic note. It means the skull itself has been shortened and reshaped, and in the boxer that shape is one of the breed's most recognized traits. What had once looked like a simple hallmark of type now appeared as evidence of a long, deliberate pressure on anatomy. This is where the ancestry begins to look less linear. The boxer was not only carrying old canine inheritance, It was carrying the visible imprint of human preference so intense that it left a broad genetic footprint. A sweep of more than 8 menobilers does not happen because a dog happened to look appealing one day. It appears when certain dogs are repeatedly chosen because they fit a desired form, and when those choices are made generation after generation, until the genome itself starts to tell the same story the eyes were already seeing. The unsettling part is how much that story rewrites the familiar image of the boxer. People usually see strength, balance, and that compact, almost sculpted head, and assume those features simply define the breed. But the chromosome 26 signal suggests a deeper process, not just inheritance, but insistence, not just survival of a trait, but its careful narrowing under selection until the line became stable enough to look inevitable. That stability is exactly what makes the discovery so revealing. It means the boxer's identity may feel natural now because selection made it that way. And once that possibility enters the picture, the next layer becomes unavoidable. If one region of the genome shows such strong pressure, what else was being preserved in silence? Which other features were shaped, protected, or amplified before anyone wrote a clear record of how it happened? The DNA has already proven that the visible dog is only part of the explanation. What comes next is the harder part, following the hidden path from selection to history, and from history to the animal standing in front of us. Then the picture shifts in a way that is hard to ignore. In 2012, a study based on genome-wide allele frequency data drew a line that earlier breed stories had never made clear. The boxer was not carrying only the legacy of domestic dogs. It was also carrying a measurable fraction of wolf ancestry that entered after domestication. About 9%, according to the analysis. Not a rumor, not a guess, but a number large enough to force a reconsideration of what the breed really is. That matters because a breed like the boxer is usually understood through the language of purpose. People see a dog built for stamina, quick response, and close work with humans, and they imagine a clean chain of breeding decisions leading straight to the animal we know today. But this result breaks that chain open. It suggests that somewhere in the long and uneven history behind the boxer, there was more than domesticated refinement. There was contact with wolves after dogs had already entered human life, and that contact left a trace strong enough to survive into a modern breed. The study did not describe a simple hybrid accident. It pointed to population splits and mixtures, the kind of deep history that does not show up in a show ring or a kennel record. It means the boxer was shaped inside a broader evolutionary traffic, where genes moved, disappeared, and resurfaced under pressures that no pedigree alone can capture. Once that is understood, the familiar boxer face starts to look less like a closed answer and more like a final surface covering older currents. And the comparison with the Basenji makes the boxer result even more revealing. The same 2012 analysis estimated about 25% wolf ancestry for the Basenji, showing that these post-domestication traces were not isolated noise. They were part of a wider pattern. The boxer sits in that pattern with its own distinct share, and that difference is important. It tells us the breed did not inherit a fixed essence from a single moment in time. It inherited layers. That is where the story becomes more unsettling than impressive. A modern breed can look stable, consistent, even carefully finished, while still carrying evidence of ancient mixing that happened long before the image became familiar. In the boxer, the athletic frame and unmistakable expression now feel less like the product of one clean design, and more like the outcome of repeated choices filtered through old ancestry. The dog in front of us is not a simple endpoint. It is a record of what survived. So the question is no longer whether the boxer has a complicated past. The evidence says it does. The harder question is why those wolf traces remained visible at all, and what they imply about the way humans shaped dogs while still being shaped, in return, by the animals they kept. The answer lies in following the mixture itself, not just the breed name, because that is where the boxer stops being a symbol and starts becoming history. In 2005, the comparison moved from theory to evidence. Researchers lined up the genome of a standard poodle beside that of a boxer, and the contrast was sharp enough to change the way the breed could be read. This was not a matter of coat, posture, or the clean confidence of a show ring stance. It was a direct look at the code underneath, the place where form begins long before a breeder can name it. The result mattered because it broke a comforting illusion. Two dogs can look equally polished, equally pure in the way people use that word, and still carry very different internal histories. According to the public account from the J. Craig Venter Institute, published under the headline, "How Do Boxers Differ from Poodles?" Researchers collar genomes. The 2005 comparison identified significant genetic differences between the standard poodle and the boxer. Those differences were not decorative. They were tied to the traits that make each breed unmistakable. That is the point where the boxer stops being only a familiar athletic shape and becomes evidence. The broad chest, the powerful head, the alert expression, the disciplined build people recognize at a glance. None of that is just surface. The comparison showed that breed identity is anchored in measurable genomic shifts, changes real enough to separate one lineage from another in a way the eye alone cannot fully explain. What looks stable from a distance is built from distinct internal instructions, and once that is clear, the story deepens in a more precise way. The boxer does not merely differ from the standard poodle because humans preferred a different silhouette. The difference is written into the breed itself, into the inherited variation that selection kept, narrowed, and repeated until it became a recognizable form. In other words, the body is not an ornament placed on top of biology. The body is the outcome of biology under pressure. That changes the emotional weight of the investigation. A breed can feel timeless when people know it through photographs, pedigrees, or the memory of a dog at their feet. Yet the 2005 genome comparison makes the boxer feel less like a fixed icon and more like a living record of choices that were anything but random. Someone wanted speed, presence, endurance, and a certain kind of balance. Generations later, the genome still carries the imprint of those decisions. The deeper surprise is that this kind of comparison does more than separate a boxer from a standard poodle. It exposes the logic of breed making itself. Once scientists could place those two genomes side by side, they could see that the differences between breeds were concrete enough to be tracked, measured, and associated with visible traits. That means the familiar category of breed is not just a label humans attach after the fact. It reflects accumulated genetic change, stabilized by selection. So the boxer's story gains another layer here. The earlier clues about ancestry and mixture no longer stand alone. They now sit beside a second truth, just as important. The breed's history is not only ancient in the hidden sense, it is also specific in the modern sense, because its defining traits can be distinguished from those of other breeds at the level of the genome itself. The boxer is not vague heritage wrapped in muscle. It is a set of differences that can be pointed to, compared, and traced. That is why the 2005 finding lands so strongly. It does not solve every mystery about the boxer's origin, but it confirms the most important one. What people see as breed character is rooted in real biological divergence, not in appearance alone. And once that door opens, the next question becomes unavoidable. If the boxer can be separated so clearly from a standard poodle, what else was preserved, hidden, or sharpened in the long path that made the breed what it is today? The next clue did not come from a grand history book or from an old kennel ledger. It came from a practical problem. Who sired which puppy, and how could anyone know for sure? In 2004, researchers turned to DNA fingerprinting using microsatellites to solve parentage testing in the boxer breed, and that shift changed the scale of the story. The question was no longer only where the boxer came from in a broad ancestral sense. It became whether genetics could identify family ties inside the breed with enough precision to settle real disputes. That matters because a parentage test is a very different kind of revelation. It does not speak in broad lineages or distant echoes. It points to a particular mother, a particular father, a particular litter, and asks the genome to answer without hesitation. In the boxer, the answer was yes. The microsatellite markers worked as a molecular identity card, separating one breeding claim from another and showing that the breed's genetic structure could be read at the level of individual relationships, not just at the level of population history. For breeders, that kind of proof is not abstract at all. A pedigree on paper can look tidy, while the biology underneath tells a messier truth. A claimed sire may be wrong. A registry line may hide an error. A litter may carry uncertainty that only shows up when the markers are checked. The 2004 study made that uncertainty visible, and then it showed a way through it. The boxer was not merely a symbol of selective breeding. It was a case where inherited markers could expose whether the story attached to a puppy matched the story written in its DNA. That is the deeper turn in this part of the investigation. The same breed that had already revealed hidden ancestry at the genomic level was now proving something even more immediate. Its family structure could be verified scientifically. This is where the boxer stops being only a subject of historical reconstruction and becomes a living system of traceable relations. The genome is no longer just a map of old mixtures. It is a tool for confirming who belongs to whom, generation by generation, inside the very population that humans thought they already understood. The PubMed record for the 2004 study places that work in plain view. DNA fingerprinting using microsatellites to solve a parentage testing in the boxer breed. The language is technical, but the implication is human. Behind every marker there is a promise that a mistaken assumption can be corrected. Behind every corrected record there is a breeder, a litter, a puppy whose identity can be protected instead of guessed. And once that happens, the breed gains a second kind of ancestry. One ancestry reaches back through selection, mixtures, and forgotten foundations. The other runs forward through verified parentage, where the boxer is not only descended from older dogs, but also connected through real, testable family lines. That dual view changes everything. It means the same DNA that exposes deep origin can also safeguard the everyday truth of reproduction, making the breed legible at both the historical and the domestic scale. So the boxer's story becomes more intimate here, almost uncomfortably so. The mystery is no longer buried only in ancient sources or broad genetic surveys. It is sitting in the kennel, in the litter, in the handwritten assumption that a pup came from a certain sire because someone said so. The microsatellite study showed that genetics could answer those questions with rigor, and that answer quietly expanded the meaning of ancestry itself. What looked at first like a method for sorting bloodlines turned into something larger, a demonstration that the boxer's identity is maintained, corrected, and sometimes rescued by the same molecular tools that once seemed reserved for distant evolutionary puzzles. That is why this finding lands with such force. It confirms the breed's history, while also revealing the fragile human choices surrounding it. And in that tension, the boxer becomes more than a case study. It becomes proof that family, in dogs as in people, can be both remembered and measured. By 2013, the boxer was no longer just a breed with a surprising backstory. It had become a lens. A genome sequencing study titled Genome Sequencing Highlights, Genes Under Selection and the Dynamic. Early history of dogs pushed the case beyond ancestry and into evolution itself, showing that the boxer could help expose genes shaped by selection during the earliest stages of dog history. That mattered because it changed the question. The mystery was no longer only who the boxer descended from, but what kind of pressures built the breed into the animal people recognize today. The logic was elegant in a way that only genetics can be. When researchers read a genome, they are not looking at one clean line of descent. They are scanning a crowded record, a palimpsest of changes preserved, because some were useful, some were tolerated, and some were driven hard into place by human choice. In that 2013 sequencing work, the boxer stood among the dogs helping scientists identify regions under selection, regions where nature and breeding had left a visible mark. Those marks did not describe a single moment of invention. They pointed to a long process, one in which early dog populations were being shaped before modern breeds were ever formalized. And that is where the boxer became unexpectedly important. Its genome was not simply confirming the familiar story of a disciplined, athletic, square-built dog. It was helping to map the deeper machinery of domestication. The study connected the breed to a broader dynamic early history of dogs, revealing that the formation of canine diversity was already underway, long before kennel clubs gave it names and standards. In other words, the boxer was not standing at the end of history. It was standing near one of its older turning points. That shift matters because it breaks a common illusion. People often imagine breeds as neat inventions, as if a human group sat down, picked a body shape, and sealed it into existence. The boxer's genetic record argues for something messier and far more revealing. Selection did not begin with the breed card. It began with practical survival, with work, with temperament, with bodies that could do a job and keep doing it. Later, people refined those bodies into a recognizable type. The genome preserves both layers at once, the ancient pressures that predate the modern breed and the later human decisions that fixed its outline. Seen this way, the boxer stops being a closed chapter and becomes evidence in a much larger investigation. The same animal that shocked researchers by carrying a deeper ancestry also helped show that dog domestication was dynamic, uneven, and full of branching paths. Some changes were locked in early, others surfaced much later, and the breed's genome made that visible without ceremony, almost like a buried map flashing for a second under light. That is why the 2013 study lands with such force. It did not just add another fact to the boxer's file, it stretched the meaning of the breed across time. It suggested that every modern boxer carries inside it a living archive of early canine history, a record of genes favored, filtered, and preserved, while dogs were becoming dogs in the human world. Once you see that, the breed's athletic frame feels different. It looks less like a tidy result and more like a surviving shape, one that remembers older demands than the ones written in today's ring. And there is something quietly unsettling in that realization. The boxer appears familiar, almost self-explanatory, until the DNA says otherwise. Then the breed begins to look like a witness, not to one origin, but to a sequence of decisions and pressures that started long before anyone called it a boxer. The surprise is not that the story is complicated. The surprise is that the complication was always there, folded into the animal itself, waiting for sequencing to make it visible. By the time the boxer's deeper ancestry starts to feel legible, another kind of record begins to surface, and it is far less romantic. It is clinical. In 2015, a pedigree-based genetic appraisal looked at arrhythmogenic right ventricular cardiomyopathy in boxers from the United Kingdom, and the question was no longer only where this breed had come from, but what had been carried forward inside its bloodline. The study followed the disease through family lines and examined the role of the striatin mutation, known as STRN, as if a hidden sentence in the breed's inheritance had finally been brought under a lamp. That matters because pedigree is not just a family tree in the sentimental sense. In a boxer, it becomes a map of risk. A line that once seemed valuable for structure, drive, and consistency can also reveal how a harmful trait stays alive when breeding decisions keep repeating the same successful shape. The dog in the ring and the dog in the clinic are not separate stories. They are the same history viewed from different angles. One shows elegance and power, the other shows what selection was never able to erase. And this is where the boxer becomes more revealing than many people expect. A breed can be standardized without being simple. It can look fixed while still carrying a layered inheritance that only appears when disease is traced backward through relatives. The 2015 appraisal did not merely name a disorder. It showed how a mutation can travel quietly through generations, hidden by the very uniformity that makes a breed recognizable. In that sense, the boxer's ancestry is not confined to ancient crosses or early selection. It also survives in the medical consequences of those choices, written into hearts, pedigrees, and the quiet calculations breeders have to make when symptoms begin to repeat. The unsettling part is how ordinary this can look from the outside. A healthy boxer may seem to confirm the breed's stability, yet a family history can tell another story entirely. One litter may hold the first warning, another may carry the same risk without any visible sign. That is why the pedigree-based approach changed the meaning of inheritance. It turned ancestry into a question of responsibility. If the same line keeps producing the same pattern, then lineage is not a relic. It is an active force, still shaping what a boxer can become and what it may be forced to endure. So the breed's true ancestry is no longer just a matter of ancient dogs and selective crossbreeding. It reaches into the present, where genetics does something more intimate and more severe. It exposes how beauty, function, and fragility can be braided together in the same family chain. The boxer's story, once thought to be about origin, becomes a story about consequence. And once consequence enters the frame, the breed looks different again, as if every confident stride is carrying a second meaning beneath it, one only the genome and the pedigree can fully confess. What the boxer looked like on the surface was never the whole story. A square head, a spring-loaded body, a gaze that seems to wait for the next command. Those traits made the breed feel familiar, almost settled, but the genome refused that simple ending. It kept pointing backward, past the polished image of the modern boxer, toward a deeper chain of choices, mixtures, and inheritances that no show ring could fully explain. By the time researchers reached the 2013 sequencing work, highlighted in Genome Sequencing Highlights Genes Under Selection and the Dynamic Early History of Dogs, the picture had already changed. That study did not treat the boxer as an isolated symbol of a recent breed standard. It used genome sequencing to show that genes under selection could still carry the imprint of the earliest phase of dog history, revealing how domestication and later breed formation were connected by more than appearance. In other words, the boxer was not just a product of recent kennel decisions. It was standing on top of a much older biological record. That matters, because selection leaves scars and signatures at the same time. It keeps some traits, reduces others, and preserves patterns that tell on the past even when written history is incomplete. In the boxer, that meant the body people admired was not the result of a single clean origin. It was the visible outcome of repeated narrowing, deliberate pairing, and the survival of features that human preference kept reinforcing across generations. The DNA did not merely confirm a family tree. It exposed the fact that the tree itself had been pruned, grafted, and redirected so many times that the original shape could only be recovered by reading the genome. And once that is understood, the breed stops looking like a finished invention. It starts to look like evidence. Each characteristic that seems stable, the strength in the chest, the athletic balance, the guarded expression, becomes a clue that selection was working with older material, not building from nothing. The boxer's true ancestry, then, is not less impressive than the popular version. It is more demanding. It asks us to accept that identity can be made from layers, ancestral canid history, selective breeding, and the choices of people who wanted a certain kind of worker, guardian, and companion all at once. That is why the genetic surprise hit so hard. Researchers were not simply discovering that the boxer had a complicated past. They were seeing that the past had been hiding in plain sight, embedded in every recognizable feature. The genome turned the breed from a fixed label into a living archive. What looked like a modern, well-defined dog became proof that modernity itself was built on older foundations, and that the lines humans draw around breeds are never as final as they seem. So the true ancestry of the boxer is not a neat origin story. It is a layered inheritance, shaped by selection so intense that it can still be read decades and centuries later. The revelation does more than correct a record. It changes the emotional weight of the breed. A boxer is no longer just an image of power and loyalty. It is the visible result of unseen decisions, preserved signals, and a genetic past that refused to disappear. And that leaves us with a broader truth that reaches beyond one breed. Whenever a history seems too tidy, DNA may be waiting to reopen it, not to destroy the story, but to make it honest.